All right, we're going to talk about transcription steps. Now, the steps of transcription are really pretty much the same as replication. You're going to have initiation. During initiation of transcription, you're going to use a different enzyme. So we are going to use transcription. We're going to use different enzymes. So we're going to use RNA polymerase. And RNA polymerase is basically the enzyme. It ends in ase. It's going to identify where to begin transcription. So during the transcription, we're going to take our DNA and we're going to make it into mRNA. The second step is we're going to do elongation. RNA polymerase, again, which is the enzyme. Is going to synthesize a strand of mRNA complementary to DNA codons, so always in sets of 3. The third thing is termination. Termination always means to stop. RNA polymerase, again, it's an enzyme, is going to stop transcription when it encounters terminators. In the DNA sequence. Now, once it hits a terminator, it's basically just going to release off of DNA. So I want to look at the steps of DNA transcription, but before I do that, I want to talk about the different enzymes. When we talk about RNA polymerase, the enzyme that makes RNA is called RNA polymerase. Well, we have 3 different RNAs. In replication, we learned that there is DNA polymerase 3 and that there's DNA polymerase 1. Well, since we're using 3 different types of RNA, I want you to know the 3 RNA polymerases. So, RNA polymerase 1 is going to transcribe rRNA, which is going to make up your ribosomes. RNA polymerase 2 is going to transcribe mRNA. So when we make mRNA, we're going to use RNA polymerase 2. And the third one, RNA polymerase 3, is going to transcribe, which means to make, tRNA. All right, when we talk about transcription, we're going to basically draw out everything that happens, but we need to understand one more thing. Transcription factors. Transcription factors is basically going to be something that is going to be helping our RNA polymerase initiate transcription. Running out of room. All right, so transcription. Initiation, it has to start, we elongate it, and then it has to terminate. Now, in the past when we did replication, we used all the enzymes. So, we know that in order to copy DNA for transcription, it has to be unzipped. Transcription is going to be taking your DNA and making a copy of only one side to produce your mRNA. So here's your DNA. Your DNA, just like in replication, has to be unzipped. So during transcription, you draw basically the replication fork, but it's not replication that's occurring. It's transcription. So you can go through and write all the same enzymes. The enzyme that unzips DNA so that transcription can occur is helicase. Helicase is always responsible for unzipping your DNA. Now, single-stranded binding protein, SSBP, is going to prevent DNA from winding up. We always read our DNA in sets of 3 called codons. And those represent our nitrogen bases. Now, transcription occurs with RNA polymerase. This is also going to occur in a 5 to 3 prime. To build new DNA, always has to be built on the 5 prime, but it is anti-parallel, so we always draw that on here. So, before we move any further, I want to go through a couple of new things. The side that we're going to use to copy of DNA is only the left side. We don't use the right side anymore. So, the strand of DNA that we're going to copy is going to be called the template strand. In DNA replication, they were both templates. So this is the strand of the double helix. Used to make RNA. This strand is not used, but we call it the coding strand. It is not used, but it is the coding strand. This is the strand of DNA. that is complementary to the template strand. Now, we've already talked about the enzyme that makes RNA as RNA polymerase. So the side that we always use is the left side because it's easier. So RNA polymerase, and during replication, RNA polymerase is also called RNA primase. But during transcription, RNA polymerase is going to build a new strand of RNA that is complementary to the template strand. And it builds in mRNA from 5' to 3' so that it is opposite of the side that it is copying. So, RNA polymerase 1 would make RNA. So this would be RNA polymerase 2 is going to build mRNA. So it looks a lot like replication. However, we only use one side. Now, transcription factors are going to help the RNA polymerase bind. And then at the end, we're going to have to terminate it. So we stop and we're going to release our mRNA. So we release our mRNA. Your mRNA is a copy of your DNA. Before your mRNA can leave the nucleus, we have to edit it. So we have to modify it. It's kind of like a rough draft. So you make sure that everything is going to be normal. So this is called your primary transcript. Before mRNA leaves the nucleus, the transcript must be modified. Now, as far as mRNA goes, we basically talk about some codons or something that are just like not even basically read. So literally, we can just make this easy and say AAA, UUU, GGG, CCC. So the first thing that we need to do is remove non-coding sequences. Non-coding sequences are called introns. So let's just say that UUU is non-coding. You have to remove non-coding sequences which are called introns. You have to literally remove them. So we modify it. Now, anything that is expressed, we keep. So sequences that we keep that will be translated and expressed into proteins are called exons. So we need to remove non-coding sequences. So let's just say your intron is your non-coding sequence. Everything else is going to be your exons. So now you've modified your mRNA and you have exons AAA, GGG, and CCC. So your mRNA is going to leave the nucleus and it is going to attach to your ribosome. Your ribosomes are your protein factories. Here's your ribosome. Your mRNA leaves the nucleus and it is going to attach to your ribosome so that it can be read. So the first one that we would read would be AAA. So it's going to be moving in this direction. And then we're going to read GGG. And then we're going to read CCC. Now the ribosome, as it reads them, is going to build the amino acids.